Hello, I know it's been a while, but tonight I'm gonna take this time off to discuss a chapter in First Aid. It's from First Aid 2017, the biochemistry section, and it has to deal with cell cycle phases. And when I was studying this, it was very I say that a lot, but most of the time when I'm studying it's very difficult to understand. But this one was quite difficult to understand. So I had to take the time out to do some diagrams so that I would better be able to understand it and therefore explain it better to my colleagues. So here I have a diagram of the cell cycle. So this is it. Now in the cell cycle we have three states. We have mitosis, and the entire orange state is interphase, where the cell spends most of its time, and most cells or some cells have a kinesin state where they do not divide or replicate or any of that good stuff. Okay, interphase can be broken down into three other smaller stages called G1, S, and G2. Right, and so mitosis can be broken down into what I like to call PMAT, prophase, metaphase, anaphase, and telophase. And kinesense is the same thing as G0, where the cell does not replicate, it doesn't divide, such as neurons. Okay, all right, so let's explain the cell cycle. Well, in the cell cycle, we have things that are called as cyclins and cyclin-dependent kinases. Now, the cyclin-dependent kinases are constitutive and inactive. So, in a normal state, they're inactive, and constitutive means that they're in a constant number. Whereas cyclins, like the name suggests, their number in the cell varies depending on the stage the cell is in. Okay, so in cyclins, we're gonna start off in the G2 phase first, because I like to start off in that area of the cell cycle because it's easier to understand everything that goes on. So in the G2 phase, we have cyclin, we have CDK1, and it's in a normal amount or in a constant amount. We have CDK1, and that's it. As G2 progresses, we have an increased buildup of cyclin B. Okay, so here we have it. We have CDK1 and cyclin B. Now they bind to form what is known as mitotic promoting factor. So as we enter G2, more cyclin B increases, right? And then it forms mitotic promoting factor. Now this mitotic promoting factor helps the cell understand or pushes the cell into mitosis. Now once the cell has been pushed into mitosis, the same CDK1 will break down the cyclins. So the cyclins will cyclin B's will decrease, okay, and the CDKs will obviously remain constant. So the cell, now the cyclin B's has been decreased, the cell will now know that they have completed mitosis. And how does the cell know that it's completed mitosis? In the cell cycle, there are various things, three important checkpoints that are there. And there's a checkpoint at the end of G1, at the end of G2, and in metaphase of mitosis right here. So remember what just happened here when we had the MPF, or the mitotic promoting factor. It caused that cell to pass its G2 checkpoint, meaning that there was no DNA damage. And remember, the checkpoints are points in the cell cycle where the cell needs to test for DNA damage. And if there's no DNA damage, this mitotic promoting factor will lead the cell into mitosis. And like I said, once the cell has completed mitosis, the CDK1 will break down the cyclin B's, and then therefore the cell knows that it's not in mitosis anymore, but it's going into another stage, which is G2. Okay, so now that we've completed G2, okay, now that we've completed G2, we're going into G1, which is growth 1. Okay, now the G's are where the cell grows, and the S is where there is DNA replication. Okay, so now that we're in G1, we need to understand something. Normally there's a transcription factor in the cell known as E2F. E2F is responsible for making a variety of proteins that are responsible for DNA replication, or it prepares the cell to go into the S phase, right? But E2F is inactive and it's blocked by retinoblastoma protein, or RB protein. So how is this RB? This RB needs to inactivate so that E2F can be freed, so that the cell can move into the S phase. So what happens is that cyclin D and CDK4 comes in. So cyclin D and CDK4 comes in and it binds to the retinoblastoma protein, and therefore it inactivates the retinoblastoma protein by phosphorylating it. Okay, so now we have a retinoblastoma protein which is phosphorylated. Now this E2F is now active, it's freed. So what happens is that it now goes onto the DNA, and it causes the transcription of various proteins important for DNA replication to start. Okay, and the two important CDKs and cyclin A that it increases is CDK2 and cyclin A. So it increases the production of CDK2 and cyclin A complex, and it causes the cell to move into G1 and into the S phase. Also, this it crosses that checkpoint, that G1 checkpoint. Now that the cell has reached into the S phase, the E2F now produces more CDK2 and cyclin E, and it tells the cell that it's now in S phase and it needs to go into the G2 phase. So this is CDK2 and cyclin E. And once we've reached G2, guess what? The cycle continues. We have CDK1 and cyclin B forming mitotic promoting factor, pushing that cell into mitosis, and once it's reached there, the CDK1 will break down the cyclin B. Okay, so that's it so far. So far we've talked about DNA, the DNA checkpoints, or the cell cycle checkpoints, and the important stuff that happens. Okay, so let's say that there was DNA damage. Okay, so there was DNA damage. Sorry, let's say there was DNA damage. That means if DNA damage is present, the cell cannot progress into the various cell cycle phases. So what happens is, as soon as there is DNA damage is recognized by our police officer, which I like to call as P53, which is our police officer, P53 will arrest the cell by using handcuffs. Okay, and what are these handcuffs? P21. Now P21 acts as handcuffs in a sense that it inhibits the cyclin dependent kinases. So if it inhibits all the cyclin dependent kinases, remember P21, sorry, if P21 inhibits all the cyclin dependent kinases, the cell cannot progress into the cell cycle phases. Okay, so thank you for listening. If you have any questions, please comment in below. I did this this representation of the cell cycle. It helped me, and I hope it could help you. Thank you. Have a good night.